So in this video, I'll be going over the structures and functions of two groups: glycolipids, which are triacylglycerol and glycerophospholipids, as well as sphingolipids, that would be sphingomyelin and glycolipids. Please keep in mind that these are just a few examples of the glycolipids and sphingolipids. This is not all of them. So let's start off with triacylglycerols. In essence, they are just a glycerol and 3 fatty acyl groups stuck together. The way that the glycerol and fatty acids combine is a dehydration reaction, which forms ester bonds. So in order to make one triacylglycerol, the OH of 3 fatty acid carbonyl groups and the H's from all the glycerol's hydroxyl groups combine and produce H2O, because the OH plus the 1H, that forms water. And this is what we call a dehydration reaction. The function of triacylglycerols is to store the fatty acids in a more stable molecule. This is what we commonly refer to as body fat, and we can tap into this body fat, this energy source, when the body is running low on nutrients and needs to use something. Fat can also serve as insulation and protection of our internal organs. So these triacylglycerols are pretty important to the function of our body. They cannot be used to form the membrane due to it not having any polar spots on the molecule. The OHs that it has, they're gone. They've turned into the fatty acid. So there's really no polarity going on. Next, glycerophospholipids. They have a glycerol backbone, just like triacylglycerols, 2 fatty acyl groups, and a phosphate group, which is usually attached to something polar. They are also formed in a dehydration reaction, and that forms ester bonds as well. So in order to form one glycerophospholipid, the OH of the 2 fatty acids and 2 of the H's from glycerol's hydroxyl groups form to make H2O, which is also a dehydration reaction. And the last OH on that glycerol, as you can see, is sort of flipped upwards, and that bonds with the phosphate group, which has that polar group as well. These are what the different phospholipases can cut, and depending on what they cut, that would represent a different signaling molecule. This structure is what we refer to when we say that the phospholipid is made up of the classic hydrophilic head and the 2 hydrophobic tails. We're talking about a glycerophospholipid. So yes, this lipid does in fact make up the majority of the phospholipid membrane. So now let's talk about the sphingolipids. They're made up of a sphingosine backbone, but you won't actually see the sphingosine in the structure of a sphingomyelin. You will see a ceramide, which is a sphingomyelin and a fatty acid combined together. So then you would take this ceramide and you would attach it to a phosphate group, which is once again usually attached to a polar group, and now you've got your sphingomyelin. These sphingomyelins are usually found in eukaryotic membranes. They provide extra structure and support. Next, we'll go on to the glycolipids. So, glycolipids are just monosaccharides, such as glucose or galactose, and these monosaccharides, the glucose and galactose usually, they act as the head group, and they are attached to a ceramide, which, remember, is just a sphingosine and a fatty acid combined together. These are commonly used for signaling and recognition, like if you've heard of the different blood types, that is due to impart these glycolipids. And so something kind of cool about these is, so cells will use sugars as molecular markers because carbohydrates can form a wide range of these unique 3D structures, and these carbohydrates have many more conformations than proteins or lipids, and all these conformations allow super precise cell recognition and signaling and also identity tagging.